Martin Universal. We welcome you. This is A-level biology, mitotic cell cycle, and this is Shedro. Come ride with me. Let's fly higher and then dissolve these problems. Okay, come on, let's do it. Meristematic stem tissues in the growing plant. We're going to be a bit fast, guys. Growing regions of plants such as root tips. 2.1 shows a section through the meristematic region of a root tip or alien separ. So that's onions. All right, do you like onions? You can see all of that. All right, I want to take time. What is this stage? A is what, B is what, C is what. Okay, we're going to find out. Let's see what they're asking us. Table 2.1 shows the number of cells in different stages of the cell cycle that we observe in sections in the meristematic regions of alien separ, A separ. All right. Okay. What do you want from us? Now this is the table, number of interface, mean, prophase, all of that. Okay. So let's ask them. Complete table 2.1 by using the letter A to show H from fig 2.1. Excuse me. To identify one cell in each stage of the cell cycle. So one cell that is in each stage. This okay. So let's go do it. Let's go back. Yeah. So starting with interface. So guys, I want you to work this out with me. Don't hide there. I can see you. Stop hiding. Do try this. Okay. So which of these cells, all right, is an interface? Okay. Which ones seem not to be dividing? Okay. Pause the video and try it. Identify interface, prophase, metaphase, anaphase, telophase. Identify them. Okay. So if you check here, you can see. Welcome back. So you see that if you look at interface, the cell that seems not to be divided, okay, is the cell is not yet dividing. All right. So that is G. Is that okay? G, can you see? The nucleus is still present. Another one is B. So G and B are in interface. How do I know? They are not divided. The chromosome has not condensed yet. The nucleus is still present. Nucleus, can see two nuclei. Okay. All right. Nucleus is present. Maybe here semi-condensed replication is taking place here, and maybe here G1 or G2. Maybe G1 here. But both of them are in interface. G and B. Right. Let's look at prophase. Yeah, prophase is, of course, A. How do I know? Chromosomes condense and become more visible. That's A. This is prophase, chromosomes condensing, right? Now, of course, metaphase is the easiest one. Look at it. Chromosomes align at the equator. That's D, right? So you see that we have mentioned that anaphase is either G, interface is either G or what? B. Okay, prophase is A, metaphase is D. All right, let's go to anaphase. Anaphase, okay, this looks like anaphase. They're moving apart. All right. Yeah, F looks like anaphase. That's anaphase. All right, which other one? E, anaphase. Okay. Yep. So C looks like late anaphase anyway. Okay, but FE. Okay, F or E, any of them. Now, what about telophase? Telophase, H, it looks like telophase, and C. So H and C. Now, how do I mean? Can you see that cell plate has started forming here? Cell plate has already formed. So the cell is about, of course, splitting into two, and that begins at late anaphase. Telophase. Okay. All right. So let's see what they got. Yes, let's see what they got. Yep. See it. Interface is B or G. All right. Prophase is A. Metaphase is D. Okay. This anaphase. This is, of course, telophase. You can cross check. All right. Let's go on, guys, to the next question. The total length of the time taken for meristematic cells of A separ to complete one cycle at 25 degrees Celsius is 12 hours. 12 hours. Okay. All right. So similar to the fig 2.1, the length of time spent in each stage of the cell cycle can be estimated. To obtain, all right, the percentage of cells in that stage, okay, in that stage is calculated. using the data in table 2.1, calculate the percentage of cells in anaphase. And then we should calculate the mean length of time. This is not term, this is time in minutes for anaphase. Okay, so let's go say show you're working. Let's go back, guys. So first of all, the percentage of cells in anaphase. So the question is, how many cells are in anaphase? This is anaphase. Pause the video and try it. So the mean length, the mean number of cells, okay, number of counting cells, okay, for anaphase is 29. So what, this is total number of cells, right? So now we know. So we now say, of course, you know how this works. Percentage, percentage is a calculation we do all the time in bio. So the number of cells in anaphase, which is 29, all right? 29. Yeah, okay. 29. Yeah, okay. Yeah, that's 29. All over 5,000, the total number of cells, all right, undergoing the cell cycle is percentage. So times 100, because it is percentage. Is that okay? So guys, get your calculator. Of course, by the time you cancel, 20 cancels here, cancel out here, right? It will be like 29 divided by 50, right? 29 divided by 50. Okay, so the answer is 0.58. So the percentage of cells in anaphase is 0.58%. Okay, yeah, 0.58%. all right, that's what we got, guys. Now let's go to the next question. The next one says that we should calculate the mean length of time. This is time. This is time, okay, not term, in minutes for anaphase, for the same anaphase. Okay, but you see, they told us that a complete, look at this, okay, to complete one cell cycle, the entire thing takes 12 hours. Okay, and we are calculating this in minutes. So 12 hours in minutes is what? Okay, so convert the hour, 12 hours to minutes, by multiplying by 60, because 1 hour is 60 minutes. So multiply by 60. What does it give us? Let's see what it gives us. So 60 times 12, 12 times 60 gives you 720. Okay, 720 minutes. All right, so 720 minutes is the total number of time for one cell cycle, everything. Okay, now the question says the mean length of time in minutes for anaphase. Anaphase, remember, we have 29 cells all over 5,000, right? This is the ratio for anaphase. Okay, so let's find with this ratio in 720. all right, 720 minutes. So get your calculator, do it. So 720, so we say 29 times 720 divided by 5,000. Yep. So we've got what? 4.18 so minutes. So that means that the cell spends only 4.18 minutes, or 4.2 minutes in anaphase. You can do it for the rest of them. Is that okay? So guys, do you understand what we did here? All right, that's how this is. So let's look at the answer here. Okay, sorry, let me clean up part of this so you can see well. Okay, let's clean here. Okay, you can see percentage of anaphase, they got 0.58. That was what we got. Okay, and then the other one, see, the same way with, so they just said that 29 of course divided by 5,000 times this. Okay, so that's 4.18. Okay, let's run off. We don't have time. 2 marks. State one event that occurs during cytokinesis in the cell cycle of plant cells, such as those shown in Fig 2.1. One event that occurs in cytokinesis. Cytokinesis is when the split into 2. So C cytokinesis, guys. Let's go back. Yeah, H is cytokinesis. Remember this is plant, okay? So one of the stages that happen is that, can you see this? Can you see this? This is cell plate formation. Alright? So one of them is cell plate forms along the equator. Cell plate is just vesicles carrying the cellulose. They align along the equator, and then cellulose is laid down. Cell wall is formed. Cellulose is laid down. Cell plate formation. Okay, and of course, another thing that happens is that organelles are shared between the two cells. All right, then cytoplasm split. All right, into 2. So look at the entire gist, guys. No time. So see what's happening here. All right, look at this. Yeah, cell plate forms across equator of cell. Cell wall forms or cellulose laid down. This is in plant cell, but if it's an animal cell, we said that we use the word, of course, cleavage furrow, okay, invaginational cell membrane, right? But this is plant cell. Cytoplasm divides into 2. Okay, idea of organelles shared. It said reject cytoplasm constraint pinching. Okay, because this doesn't happen in plant cell. It happens in animal cell. Separates into 2 daughter cells. Ignore it. Okay. Event at late telophase. Ignore it. Organelles shared out. Alright, shared out. Cell plate formation. Vesicles transported to the equators involve cytoskeleton structures. Alright, so that is how it is. Let's run. As part of a study, the mitotic cell cycle of a student made stained sections of a root tip of onions. Allium separate this same onion and observed them with a light microscope. Okay, the student made drawings of 6 of the cells A to F using high power of the microscope. That's it, guys. See? So which stage is this? Which stage? Okay, what stage are they in? Try it. Alright, it says complete table 6.1 to show the sequence of stages in mitotic cell cycle using letters A to F as shown in Fig 6.1. So this is Fig 6.1. All right, what is the sequence? So cell number 1, cell A is the first sequence. Let's go back and follow it. All right, so A is the first sequence. This is, pause the video and try it. I'm not going to solve this for you if you don't try. Try it, please. Exam is on Tuesday. Try it, okay? Yeah, so don't forget that this is interphase. After interphase, the next thing that happens is prophase. Okay? So this is 1, remember? Let's follow it. After interphase, the next is prophase. Okay? If you remember the cell cycle, okay? This is mitosis, right? Okay, that's cytokinesis. The rest is interphase. Everything here is interphase, all right? Everything here is interphase. So this is 1, A is 1. The second one, when we come to prophase, to metaphase, mitosis. Mitosis, remember, it has 4 stages. We call it PMAT. PMAT. Okay, MAT. That's the acronym you can use. PMAT. So the entire thing here is interphase. So after interphase, the next is prophase. After prophase, the next is metaphase, M, and then the next is anaphase, A, and then the last one is telophase, which is this guy, T. Right? So this is 1. What A is prophase? Prophase is C2, because chromosomes are condensing. The next one is metaphase. They align at equator, E3. Is that okay? Yeah. Where are they separating? There's one as D, which is anaphase. Anaphase is number 4. Okay, 4 is anaphase. Okay, telophase is, can you see nuclear membrane forming back? That's 5. B is telophase. And then the cell split into 2. F, that's 6th event. Okay. All right. So, no time. Hope you got it. Interphase 1. Prophase 2, metaphase 3, anaphase 4, telophase 5, and then cytokinesis 6. Okay, so let's see what they've got there. See, So the same thing with that, right? So you can check it out. One mark, just one mark for all of this work. That's too much. Table 6.1 shows some events that occur during the mitotic cell cycle in A-cepa. Onions, very good it has antimicrobial bacterial activity. Okay, complete table 6.2 by naming the stage of the cell cycle when each event occurs. Oh my gosh. Okay, now pause this video and try it. In which stage does DNA replication occurs? Okay, pause the video and try it. Of course, this is interphase. That's the S phase of interphase. All right. Yes, division of centromeres. That happens in anaphase. Okay, when the sister chromatids are separating. Condensation of chromatin, prophase. Contraction of spindles. All right, contraction of spindles occurs in anaphase. All right. Okay, so if you check it out, so you have DNA replication interphase or S phase. Okay, anaphase, prophase, anaphase again. Okay, 4 marks. Explain the importance of mitosis in the immune response. Now, what is the importance of mitosis in the immune response? Pause the video and try. Okay, pause the video and try. So mitosis is involved in clonal expansion. Okay, where the lymphocytes divide to form identity. Remember, every time mitosis is mentioned, you must add genetically identical daughter cells, right? So where lymphocytes divide to form genetically identical daughter cells. So mitosis helps to increase the number of the lymphocytes when they undergo division by mitosis. And of course, they said explain the importance. The importance is that of course it forms genetically identical daughter cells. So if it is B lymphocyte, it will increase the number of B lymphocytes, so we have more number of plasma cells which make larger quantities of antibodies. Okay, if it's T helper, so we have more number of T helper cells which will make more cytokines that will stimulate more B lymphocytes, more macrophages and T killer. If it is T killer will increase the number and destroy more infected cells and pathogens. Is that okay? So that is it, guys. Right? See the importance. It forms clones. That is it. It helps to form clone. Clone is genetically identical. So mitosis helps to form clones, and clones of any of these B or T. Okay, consists of very few, but mitosis increases the number. Right? So mitosis occurs during clonal expansion, is where it occurs. Many B cells or plasma cells to make antibodies. Okay, this is many, not may. Many T helper cells to secrete cytokines, all right, lymphokines or interleukins. Many killer cells kill infected cells. Can you see that? Yeah, I didn't mention memory. Many memory cells are formed. Memory cells are produced for secondary. Yes, they are produced for secondary response, faster response when antigen encounters again. Okay, during a secondary infection. Okay. All right, see, very cheap. Fig 3.1 shows the nematode worm Canohabithes elegans. Okay, elegance. It looks so elegant. How can a worm be this elegant? Elegant worm. Okay. Now, this is the stem cells of C. elegans have been studied. Fig 3.2 shows change in mass of DNA per nucleus in stem cells during one cell cycle. Okay, let's see it. Okay, that's mass of DNA, right? As time goes on during division, you can see how it changes. All right, okay. So what do they want from us? State the phase of interphase shown by A. This is A. Can you see? And B. So you see that A, the mass is half. Okay, from here to here. Is that okay? Yeah, it's from here to here is double. Before then it's doubled. So can you see when it reaches double, it now splits again. So this is half of that. That's the original diploid number. This is when it doubles. So this is G1. Okay, this is G1 growth phase 1. The cell is young, so it's still got the original diploid number. Okay. Yeah, that's G1. Should I show you how? Okay, all right. If you check this, okay, look, let me just show you something, and then we go. Now this cell is young, and then you see the cell it's very young form. So that's G1. Okay. All right. And everyone was asking this, what do you want to be when you grow up? He said, I want to be like my mommy. Okay. See what is happening here? The cell is growing larger, and then of course it grows in size, so doubles the genetic material is doubled now. Can you see? Now, of course, the next thing that happens, I'm going to show you. Nuclear division occurs. You can see that. Right? This is mitosis. Right? The cell is undergoing all of these stages because you just want to be like the mommy. So next thing that happens is that the cell starts splitting into two. Right? Oh, sorry. That's two somehow. Okay, let's clean it. Let's clean this. Yeah, make it better. So guys, so this is cytokinesis occurring. The cell, right, pinches inwards. Okay. Yeah. So you can see. So this is cytokinesis. The cell is undergoing cytokinesis here. Right? Can you see? And then it forms this, and then the journey continues. It's a cycle. So the point I'm trying to make here, guys, is that at this point, can you see the genetic material is inside is just normal size. So this is G1. A is G1, right? B is, can you see that the mass of DNA starts increasing, and that happens during S phase of interphase. Okay, because it helps to replicate DNA is increasing. Okay, so if you check now, see, that's it. So this is G1, okay, and S phase synthesis, right? DNA replication is occurring. Okay, so that's 2 marks. That's a lot. State the stage of mitosis shown by D in figure 3.1. Look at it. So if this is G1, okay, I want you to pause the video and try it. Remember how we roll. Remember how we roll. This G1, this is S phase, and you know how we do our things, guys. We solve it. Everybody is involved. Okay, get into the game. So this is mitosis, and mitosis has prophase. You know it. Prophase, metaphase. Okay, PMAT. That's a math. How can somebody answer PMAT? All right, that's an acronym. So this is cytokinesis quickly, right? And that is G1, okay, and that is S phase. That's okay, S, and that's G2, guys. So you can see. So this should be, we have G2 here. G2 is part of here. Okay, the DNA is doubled. And part of it here is prophase, part is metaphase, right? And then anaphase. When we came here, the cell starts separating. So, and of course, the stage before cytokinesis is going to be telophase, right? So these should be telophase. How do we know? We follow the diagram, okay? Okay, look at it, guys. Prophase, metaphase, anaphase, telophase. Telophase is before cytokinesis, and this is cytokinesis. So this stage is telophase, and of course, the cell is already splitting. Okay. All right. The daughter nuclei have divided, so this is telophase. All right. Okay. So that's telophase. 1 mark. Outline what happens in a cell in preparation for cytokinesis. If a cell is preparing for cytokinesis, I want you to write down what do you think happens. If this cell is preparing, first of all, the organelles. Pause the video and try it. Pause the video and try it. The organelles are shared out between the between the poles. Okay, 2 halves. Remember, the organelles are shared out, but this is an animal, right? So the next thing that happens is that, of course, don't forget that this is important because the organelles are shared out. The next thing that happens is that there can be cleavage furrow. Okay, cleavage furrow is that invagination. You know that vagination process. Okay, when this is folding inward, can you see the cell is folded? That's called invagination. This cell undergoes, okay, folds inward. right? So the cell has to increase in size before it divides more cytoplasm, share out the organelles, then cleavage furrow occurs. Cleavage furrow, where is it? So it's folding. This is a furrow, cleavage furrow, right? That's invagination. Okay, increase in size. Let's see a catalog of that. Increase in cytoplasm, okay, organelles increase. Nuclear envelope, yes, I didn't mention it. Nuclear envelope forms around each chromosome, yes. That happens at late, of course, telophase. Alright, look at the spindle, this is, it breaks down, cytoplasm, pinches, alright, in folds, cell membrane is folding in, okay, alright, around the equator, okay. Yeah, let's post 2 marks. A stem cell of C. elegans has 12 chromosomes. Complete table 3.1 to show the number of nuclei within the stem cell in stages A, B, D of the cell cycle, shown in figure 3.2, and number of chromosomes in each nucleus during these stages. Okay, so remember what we call this. This is A is what? Z is G1, right? B is S phase, right? And D is telophase. Let's go back and check again. That's what we called it, right? It's not what we called it. Yeah. We say that A is G1, B is S phase, D is telophase, right? Okay, telophase is something like this. Can you see? So in G1 and S phase is still one cell. Is that okay? Alright, one cell. Okay, the chromosome number has not doubled. So in S phase, in G1 it has not doubled. S phase is still doubling, but has not doubled. Okay, so we still have one cell here, one cell here, one cell here. So in T telophase, alright, we still have one cell but 2 nuclei. Is that okay? In T telophase, alright. So if you go back to that question, guys, to look at what they're asking us to do, what are they asking us to do? Alright, so 12 is the diploid number, right? The number of nuclei within the stem here is one nucleus. This is 2. Okay, number of chromosomes in each nucleus will still be 12 in each nucleus, right? Each nucleus will contain, right, 12. All right. So but if they said in one cell, this could have gotten 24 because there are two nuclei here, right? But in each of them has 2 nuclei, each of them has 12. Okay, all right. So see that young nematodas have stem cells throughout the body. These will summarize the 3 cell cycles of one of these stem cells. Okay, so you see what's happening in the stem cells, dividing to form other cells, stem cells, and other cells that differentiate. Okay, so with reference to figure 3.1, outline the role of stem cells in animals. So what is the role of stem cell in animal? So the role, pause the video and try it. Okay, yeah, so stem cells, you know that they divide by mitosis continuously to produce other cells for growth, for repair of tissues, or or replacement of cells. Okay, and then other cells that also differentiate. Look at it, guys, memorize it. This question is always coming out. Stem cells continue to divide. They divide repeatedly by mitosis, you must mention it, right? And they form genetically identical cells. Why? For growth, for repair of tissues. Don't say repair of cells. Cells are not repaired. You say replacement of cells. Okay, a replacement of worn out old dead cells, damaged cells if repair of tissue is not given, to differentiate for formation of tissues and organs specialized. Okay, now also it also forms, all right, other cells that continue to divide. Now the potency are multipotent, pluripotent. So as stem cells differentiate, okay, their potency changes, but there are 3 types: multipotent, pluripotent. It's more potent than this, right? Okay, 3 marks. Now the early development of an animal involves division of the zygote and daughter cells by mitosis to form embryo consisting of genetically identical cells. Fig 4.1 shows several cells at various stages of cell cycle in an embryo of whitefish courageous arctidae. Okay, what stage is this? What stage are they? Microtubules. Name the stage of mitosis in cell A and in cell B. It's simple. A, look at it. Can you see the one that is visible, that stains, is chromosome. Now, chromosomes, pause the video and try it. Can you see that instead A, chromosomes line up at the equator? This is a cell. Can you see these are the microtubules, spindles? Now these lines are at the equator, at the middle of these poles. That this is a metaphase. Okay. Now cell B, where are you? Can see that chromosome they have started dividing. They started moving to opposite pole. So this should be anaphase. Okay. Yeah, anaphase. A, metaphase, B, anaphase. Fig 4.1 shows microtubules in the cells that are dividing. Describe the role of microtubules in mitosis. Okay, mitosis has 4 stages: prophase, metaphase, anaphase, telophase. Okay, what are the roles of microtubules? Okay, it starts forming in prophase, but in metaphase, what is the role? The role is that it attaches the chromosomes, right? Attaches to the chromosomes, the centromere, and then helps to align them at the equator in metaphase. In telophase, all right, remember that it decondense, microtubules, they polymerize or shorten at the poles in order to pull sister chromatids apart. Okay, in telophase they disintegrate and then push the 2 nuclei apart. Look at it, guys. They're always asking this question. They form spindle apparatus attached. Okay, late prophase, that's when they're full. Okay, start forming sister chromatids align at the equator. Metaphase, during metaphase you can see, until anaphase maintain sister chromatids. Okay, and anaphase they shorten and then centromere divides, but they help chromatids move to the opposite pole. So daughter nuclei of one, okay, chromatid of each chromosomes. Daughter nuclei receive a full complement of chromosomes. So the poles are lengthened to extend the cell. So it pushes it, daughter nuclei apart in telophase. All right. Okay. So what happens in cell C, shown in figure, until 2 new cells are formed? Right. This is talking about cytokinesis. What happens? So pause the video and try it. What happens? Right. 2 new cells are formed. State what happens in cell C, shown in fig 4.1. What happens in this cell? Okay, cell C. So what is cell C doing? Cell form. Okay, what happens? Okay, what stage is this cell? Can you see? This is cell C. This is already telophase. Right, telophase. That means that what happens is that, can you see that? Can you see there's invagination here? The cytoplasm, the cell membrane folds inward. Okay. Okay, remember that nuclear membrane will be formed around each of these chromosomes, sister chromatids. So nuclear membrane forms. Okay, spindle disintegrates. Organelles are shared out. Right. Cleavage fluid forms. All right. Let's see what I got. See what I got here. All right. So nuclear membrane reforms around each group of chromosomes. Yes, the chromosomes. That these are features of telophase they're asking you, and into, of course, cytokinesis. Uncoiling in telophase, then coil, nucleolus reforms, reappears. Yeah, organelles distributed between 2 halves. Cytokinesis. Okay, divisions, cleavage, fluoroforms. Right? Yeah, pinches from the equator. Please memorize this. 3 marks. The cells produced in developing whitefish are genetically identical. Identify and explain, okay, 2 events that occur during the cell cycle that lead to daughter cells being genetically identical. Try this. So what are the events that occur that causes cells to become genetically identical? Guys, in cell cycle. Pause this, try this, okay? Pause it. Now I think that the first one is semi-conservative replication of DNA that occurs in S phase, okay? Alright, so that causes genetically identical cells. It's part of it. Then the second one is during metaphase, the chromosomes align at the equator. Okay, but don't forget that that semi-conservative replication of DNA produces chromatids. Alright, I'm just trying to do something here. Okay, so because of semi-conservative replication of DNA, watch. So we now have sister chromatids. So these 2 sister chromatids formed joined by centromere. So these chromatids are genetically identical. Alright? And then at metaphase, can you see? At metaphase, this is another event. You know, at metaphase, then we have spindle fully formed, right? And then they are now attaching. They are attaching at spindle. Okay, so you can see this same chromosome. I'm just using it. Lines up. Okay, so the orientation of chromosomes, all right, ensures that each, all right, daughter cell each have received, okay, the sister chromosome. So this will move to this pole, this will move to this pole, and at anaphase they divide. So its anaphase also helps, because anaphase, each chromatid, sister chromatids, they are pulled to the opposite poles. So I've mentioned semiconductor replication of DNA. I've mentioned, of course, metaphase, that they are aligned in such a way that they can be separated, and I've mentioned anaphase that allows them to be pulled apart to opposite pole. All right, look at it, guys. Semiconductor replication of DNA is the first one. Idea that each new chromatid is identical, all right, to old one. So this is identical to this. This is used to make this body identical. Right. Each chromosome has two identical DNA molecules. Yes, alignment of chromosomes on the equator at metaphase plate. Can you see that? So why? Because chromatids separate, so each cell receives a sister chromatid. So when they separate, each cell will receive it. Okay, centromere split, shortening of, all right, all of that helps to separate them. Checkpoint occurs within cell cycle to ensure that, you know, that there are no genetic errors, so they are genetically the same. All right, so there's no variation. So that errors in replication are repaired by DNA, right, to avoid mutation. So memorize this, guys. This is the concept, but you've got to use the word of the math scheme. Okay, if you want to excel in the exam, use the words of the math scheme. During interphase and mitosis of the cell cycle, interphase and what? Mitosis. Interphase and mitosis. Yeah, the chromosomes within a cell go through a number of changes. Each chromosome is composed of DNA complex with protein. Okay. In interphase, individual chromosomes are too diffuse, long and thin to be visible using a microscope. In this stage, the chromosomal material is known as chromatin. State the term used to describe the protein that are complex with DNA to form chromatin. So which protein combines with DNA to form chromatin? Okay? Yeah, of course it's histone protein. Histone, that's one mark. When viewed through a microscope, a chromosome is most clearly visible during the metaphase stage of mitosis. Okay, Fig 1.1 to produce a labeled, or let's say it's insert to produce a label diagram of the metaphase stage of mitosis in an animal cell with two chromosomes. Okay, two chromosomes at metaphase. Pause this video. Okay, draw two chromosomes at metaphase. That's what I want you to do. All right. Yeah, this is the pose. You know how this works. And then of course we have the spindle running between the poles. Okay. Yes, there are many of them. Okay. The aster. Okay. Yeah. So of course many of them, but I'm gonna use just two. Right. So chromosomes line up along the equator. Two chromosomes. This is the first one. Right. So this is sister chromatid joint under sister chromatid attached to the spindle by this kinetochore, the centromere. You can see aligning at the equator. This is the second chromosome here. Okay, the sister chromatid. All right, this is all they wanted to do, guys. Okay, yeah, show that you walk away with your marks. Okay, all right, that's it. But if they wanted to show that of my what is it called, anaphase, you have to show a V shape, right, showing it moving to the opposite pole, right? That's how the chromatids will appear, right, attached to this is the pole here, right? This is that for anaphase, right? Because questions come like that for anaphase, all right? But for metaphase, that's just going to be, right? Okay, look at what they said here. Two chromosomes, each of two chromatids drawn opposite along the central equator. Here, that's what we've done now. Plus one drawn feature. Now they want us to label. You can label it. This is centromere, sister chromatid. This is spindle, right? This is spindle. That's centromere joining them. Okay, these are two sister chromatids, right? And all of that align up at the equator. Okay, outline the changes that occur to the structure and behavior of a chromosome from the start of S phase to the end of interphase. Yeah, so start of S phase, pause the video, because mitosis too, the chromosomes undergo, of course, the DNA undergoes semi-concentric replication. All right, that's what happens, and then of course checkpoint and all of that. But in prophase, because look at mitosis, chromosomes condense. Anaphase, they are aligned at equator. Prophase, they separate. Okay, all right. Of course, they decondense at telophase. Look at this. Okay, chromosomes then replicate, DNA synthesis, which is stated in. So this doesn't occur. Replication does not occur in G1, G2. Each new molecule then complexes a histone protein. So you see prophase, what's happening here. Okay, become shorter, coil, become visible up here, become attached to sister chromatids. All of those things we've been learning in this video. So we have done a lot in this video. So I want you to, solve more questions. Okay, play the video, share with your friends. So thank you for staying true. This is Shedro. Bye.